Thank you very much, Brian, for actually twisting my arm to come to this conference. I won't say I was reluctant, it was just that I had never been to an EHC conference, and I saw it was in the far north in October, and I thought we'd be in log cabins and it would be almost dark all the time, but of course I was completely wrong, and here we are in this beautiful sophisticated hotel, and it's sunny and warmer than London was when I left. The topic of gene therapy has been one that I've pursued for nearly 16 years now. First got really interested into it back at the turn of the millennium, and I was fortunate that one of my PhD students, Amit Nathwani, when he left me, went off to St. Jude and worked with the team there, Nienhuis and David Off and others, towards developing an AAV vector. And his first target was sickle cell disease, but they quickly learned that even if you could transfect a bone marrow stem cell, the AAV transferred gene would not persist in the generations of daughter cells, and so they switched to haemophilia B factor 9 as an easier target. So that's how he got to be working. And then when he came back to London in 2004, he continued that work at UCL. At that time, I was still working with the Medical Research Council at the Clinical Science Centre, but an opportunity came to return to the Royal Free, and I went back there in 2006, actually specifically, as I said at my employment interview, to initiate gene therapy studies, and that's what we were able to do. And I'm going to tell you about that study, which started in 2010, it's been published twice and is still ongoing, is the first successful long-term success in gene therapy for haemophilia, and that has opened a certain door and in fact almost a rush, and there's now a lot of investment from industry into gene therapy projects, some of which have been recently announced, World Federation of Haemophilia, and I think now we're learning a lot more and a lot more quickly. So there's much more to say than, for example, when I was giving talks on gene therapy over the last decade. So with that introduction, I'll just give you my, I thought I'd add that because we're in Norway. It surprised some of my colleagues, but it was 50-50. It was a close-run thing, and the UK decided the way it decided. I have two company involvements to mention. We have set up at the Royal Free with Wellcome Trust funding, a academic industrial spin-out called Freeline. And I'm also working with our wonderful colleagues at BioMarin on the haemophilia A gene therapy study. Well, what do we know about current treatment? You've been hearing it's safe and it's effective, and there's exciting new advances that Johannes is going to talk about after me. What's special about gene therapy? Well, it's a potential for cure by continuous endogenous expression at therapeutic levels after a single dose administration. Haemophilia is a very good target for a gene therapy approach. Single gene defects, the genotype-phenotype correlation is generally very good. The therapeutic goal is fairly modest. An increase to 5% completely changes the bleeding frequency. 12 or 15% eliminates spontaneous bleeding into joints completely. As we know from the experiment of nature, which is mild haemophilia, as that wonderful paper from Den Uyl showed us so clearly. And we've got a very easy, quick way to validate efficacy immediately. Tight regulation, not necessarily required, but excessively high levels could be a problem, and we have to bear that in mind, as indeed we've already seen some very, very impressive levels in one of the gene therapy studies, but not threatening in any way, but it's something we must bear in mind ultimately. The popular vector of the jour is the adeno-associated virus. They've got a very good safety profile. They're not pathogenic. It's a single-stranded DNA. It'll only replicate in the presence of helper virus, for example, the adenovirus, the common cold. The technology for manipulating this virus has been developing for over 30 years now, so it is possible to engineer the capsid and to entirely engineer the expression cassette, in other words, what you replace the viral DNA with. We've known now from many years' worth of experiments in animal models that a single dose administration with tropism to the liver will result in long-term stable expression, and we're talking about 15 years in dogs, 10 years in monkeys, lifetime in mouse. There's many serotypes that have got distinct tissue tropisms to liver or heart or brain, and they can be manipulated. So we take out the viral DNA with its instructions for making the necessary proteins to make more virus, and we put in our chosen DNA instruction, for example, the cDNA of factor 9, with an appropriate tissue-specific promoter, for example, LP1 is liver-specific, and polyadenylation. We have to leave in the inverted terminal repeats because they're necessary for packaging. They do, in principle, enable some degree of integration, albeit at a low non-specific level, and we can return to that if we discuss long-term safety issues. We can pseudotype, that is, we take the DNA from one strain of AAV and put it in the capsid of another, and that's what we did with this particular expression cassette. We used another technical trick in a way here with the expression cassette in our first factor 9 study. It's a self-complementary, and what this enables is that upon release of this single-stranded sense, antisense, opposite direction piece of DNA, that it will then fold back on itself through Watson-Crick pairing and form a double-stranded DNA. The reason why that's advantageous is that single-stranded DNA is a target for DNA nucleases, which is one level of viral defense that our cells have, and which limits efficacy of single-stranded DNA, albeit not perhaps as much as we originally thought, but it helped. So the objective of our study was a phase 1/2, assess safety, efficacy of a single bolus infusion into a peripheral vein. We were able to move on from Kathy High's earlier study, which was the study we built this design on, and by using the tropism of AAV8, which is highly tropic towards the liver, at least in mice, perhaps less so in humans, we could get away from her direct infusion approach using an AAV2 capsid, which she had to infuse into the hepatic artery. So we could give in a peripheral vein and still get most of the capsid delivering the expression cassette DNA into the liver. We didn't give upfront immunosuppression. Why would we consider such a thing? Because the two subjects given that high dose in Kathy's study that was published in 2006 both developed an immune hepatitis based most likely on T-cell attack on liver cells that were displaying the AAV2 capsid peptides. Our entry criteria were no pre-existing immunity to AAV8. These viruses do circulate in the community. Everyone here has probably been infected with AAV2 in childhood. As I said, it's non-pathogenic. There's no symptomatology or pathology associated with wild-type adeno-associated virus infection. So that safety has to be our highest criteria in any development of a treatment, especially for a condition where there's already safe, effective therapy. We had a very elaborate informed consent process, which amounted to full engagement with the volunteers in understanding as much as we knew about the virus and its effects or potential risks, up to and including death. Our first patient is a very brave and completely altruistic in his motivation. By the way, he's Polish. And he'd come to London, and he said, well, you've helped me a lot and put me on prophylaxis, and I'd like to give you something back. Yes, I'll take part in the study. And he received a dose which we did not expect to give rise to an expression level that would make a difference to his condition. So he had this first infusion at a dose level, which I'll mention in a moment. Yes, 2 × 10⁻11. This is just following the vital signs. We did not then, or in any subsequent infusion of this vector, observe any change in vital signs. So there's no immediate reaction at all. And although we've been giving these infusions as inpatient, in fact, we plan to give them as outpatient in future. Vector shedding, we're giving a high dose of vector, and you can detect it using a PCR for the expression cassette out to a number of days in different body fluids. We're particularly concerned about semen, and so we do have to require that subjects agree to barrier contraception until we've got negative readout on successive semen samples. However, it does disappear all over a period of 2 or 3 or 4 weeks. So this is to give more details on the subject. 2 × 10⁻11 vector genomes per kilogram. So that's up in the billions of individual particles. I mean, it still looks like a clear fluid. It does, of course, take quite a lot of manufacturing effort to make this large number of vector particles. He has a missense mutation. He had been on twice-weekly prophylaxis with a fairly low breakthrough rate. The level at the beginning is due to his previous prophylaxis. It fell during the first week to 2%. It has never fallen below 2%. Remember, his baseline level is less than 1% in the subsequent 6.5 years. So this is the longest running observation on an individual who's received gene transfer for haemophilia B. So there was a detectable expression level, and we observed a change in phenotype. He's been able to stop his prophylaxis. He still has occasional, always trauma-related bleeding for which he receives on-demand prophylaxis. And there's been an observable change. This, obviously, these slides have to be updated. That should say more than 6.5 years without spontaneous hemorrhage and without prophylaxis. We then moved on to treat another patient at the same dose. We had our plan for the study was that if each of 2 subjects at a given dose level did not attain a steady state level above 3%, then we'd proceed up to the next dose level. Next subject, more severely affected with a null mutation, older, very damaged joints. He too, totally altruistically motivated, said, fine, I want to just help you out with this study. He did, however, get about a 1% level. We were able to establish this by, he himself wanted to know what it was and went for 3 weeks without any treatment and then had a bleed. But his level had gone to about 1%. We have been able to change his prophylactic regime to once a week from twice a week, and he's doing well on that. So it somewhat, perhaps debatably, has made a difference to his treatment regime. We then moved to the next dose level, 6 × 10⁻11 vector genomes per kilogram. After that, this subject did achieve, slightly disappointingly to us, considering we'd gone up 3-fold, only about a 2% level. He's back on prophylaxis, albeit with a slightly prolonged interval. We were able then to, we treated a 4th subject at this same dose level. He did slightly better at that dose level. He had a few traumatic bleeds early on, but his level has settled out at between 3 and 4%. And that has continued for now more than nearly 5 years. He's off prophylaxis. He does play. He's a very keen sportsman. He's in. He returned to his native South Africa, and Johnny Malanga looks after him now, so we get regular updates from him. However, because we didn't get both subjects above 3%, we then moved on to the next dose level of 2 times 10 to the 12, high dose level, and this subject was the first subject in whom we saw the transaminitis developing. Now, what we're tracking here is factor IX level and the ALT level, which is sensitive to this complication occurring, the one that had been seen in the earlier study by Kathy High. By the way, you never see this in animal models at all, at any dose. It's specific to the human immune response. And at week 7, 8, he developed a sharply rising ALT and a falling level of factor IX. It had been going along nicely, about 6%, and we then instituted what we'd planned to do, which was a short course of steroid. The ALT rapidly came down. Other evidence of liver inflammation disappeared, has not recurred at any point, but he lost some of that expression, no doubt because this T-cell response is targeting the cells that are expressing the most capsid peptide, and therefore the ones that are likely producing most of the factor IX. So he has ended up with a factor level that hovers around 2%, and he is back on a prophylactic regime. Albeit at a somewhat diminished or extended frequency. At 6 weeks, in other words, before that had happened, I'd started the next patient. So you can imagine I was a little bit anxious at this point in time. And sure enough, this subject, whose factor level from that point was clearly maintaining around 8%, then got a very small rise, but we were watching very, very carefully now every other day, ALT, and as soon as we saw that little rise and that fall of factor IX, we gave him a course of steroid. That sorted it out. He's now running a factor IX level at around 5%. He's off prophylaxis now at just under 5 years. He hasn't had any more on prophylaxis or on-demand treatment for bleeding in that time, and he's again, we seem to pick very sporty patients, but he's a very keen sportsman, plays a lot of football, runs marathons, and he's been doing all that now for 5 years without taking any factor IX concentrate. So to summarize this first stage of the study, we've had 6 subjects. We've got evidence of sustained level of expression. Just update that to over 5 years, almost 6 years. And half of these patients have off prophylaxis. The other 3 have at least changed the prophylactic regime. And we, however, have seen this immune response to the AAV capsid at the high dose level, but we can control it with a short course of prednisolone, but there's no late reoccurrence of liver inflammation. We recruited another 4 subjects in 2012 to address these questions. In the interest of time, I'll move on to the actual data. The subject 7 with a null mutation, he attained a level of factor IX over 5% with a very good result. Subject 8 did not develop any immune response at all, unlike subject 7, who got a response and had some steroid, an immune reaction apparently there. He's done very well. His factor IX level stays around the 6-7%. This subject, rather similar, no immune reaction, and a factor IX level around 6%, which is holding up well. And the 10th patient did get an immune reaction rather more prominent, and he lost some expression level, and he's running along about 2%. So to just give the headline on that, the annualized bleeding rates have gone down, as have the factor IX concentrate usage, more so for the high-dose patients. And there's a lot of saving at the current prices of factor IX. And to summarize, we've got reasonably consistent expression at the high dose level, around 5%, especially if there's no or one can rapidly eliminate the immune reaction to the vector capsid, and many of these patients can stop prophylaxis. We have got this immune response, but it does respond to prompt steroid treatment. Now, other trials have been evaluating similar vectors, and I'll say a little bit about them. This is a table that was put together by Glenn Pierce and lists the various gene therapy studies, preclinical and clinical, ongoing now, and recruiting and reporting are the SPARK study, which I'll just say something about, the Unicure study, and these are in the planning stages. This, I think, is a very interesting data from Kathy High's group, and here they're using a vector into which they've incorporated the gain-of-function Padua mutation. They've chosen a dose, which seems to be a very happy choice, at which they're getting a good level of expression, but without a transaminase response. They're not giving immune suppression, and as at the announcement of follow-up data in Orlando in August, these subjects are maintaining factor IX levels after a single dose of this vector out at beyond 4 months, and without any evidence of a immune reaction. They've not required factor IX replacement therapy. These levels are actually what you would predict if in our study we'd used the Padua gain-of-function mutant in our expression cassette. So it's very consistent with our studies at our intermediate dose level. So you multiply our levels by about 6 or 7 and you get this kind of level in activity terms. The haemophilia A was considered perhaps a leap too far, given the problems one might have in fitting this very large cDNA, 7 or 8 kb into AAV. Obviously not possible, but fortunately we can check out most of the B domain, as has been done with the Refacto, the protein that we use. And a number of studies are planned or actually in progress. Now, the first one that is in progress is the Biogen study, is the BioMarin study, which has been announced. And we now have reports on, I think, 9 subjects have been treated, 6 subjects have got good levels of sustained activity. And the plan has there been to use, after seeing an immune response at an intermediate dose level, to put all patients on upfront steroid to suppress immune response. Now, you can see there's a lot of people have now got a plan to come in here with various approaches, including the zinc finger nucleases from Sangamo, probably Caspar will get in there soon. Lentivirus vectors are also under consideration. This is the sort of summary slide that we saw in Orlando, and which is available for you to look at on BioMarin's website. What we see is at the high dose infusion, we see that is 6 times 10 to the 13 vector genomes per kilogram. This is a codon-optimized B-domain deleted factor VIII cassette that we had studied in animal models, and here in the human there's a very good expression level, even up into a slightly high level, not quite super physiological, because some people walk around quite happily with a level of factor VIII in that sort of range. And this has been well maintained since infusion out to, I believe, 4 or 5 months now. So this is very encouraging. The kinetics, there is also some slight elevation of ALT, which persists for a period and then disappears. The conclusion on that is that a single or rather these are my overall conclusion, that with AAV-mediated gene transfer, we've established a new paradigm. A single administration of vector can give long-term expression. We've seen that out to well over 6 years. That there's a corresponding, as you would predict from the plasma levels, reduction of spontaneous bleeding, clotting factor usage, and the patients on an anecdotal, individual level report, as far as they're concerned, it's a very marked improvement in their quality of life. So the Padua gain-of-function mutant in factor 9 has now pushed levels attainable, and this has definitely raised the bar to what we should be aiming for, up into the high mild range, not quite the normal range. And factor 8 activity levels well into the normal range have been now obtained, albeit with a steroid administration prophylactically. So I think that the era of cure of haemophilia by gene transfer no longer seems an unattainable vision, but one that's within grasp. But we do have many questions still to answer. And no doubt we will take those up in future trials and perhaps in our discussions. The key ones are how long will it last? And I would say, is there really any kind of long-term risk from insertional mutagenesis? And these will feed strongly into the point at which we start addressing whether and when to treat children. So, thank you.